Hello everybody, welcome to HITS. My name is Walter Parson. I'm talking to you from Austria. And today I would like to share two cases with you where we use massively parallel sequencing in forensic genetics. Now the first case is a case that goes back to the beginning of the 19th century. And we were investigating whether we could identify the DNA in bones that have been macerated. As you know, maceration is a very strong process that destroys DNA. And in this particular case, that was the challenge. We are talking about a person with the name Johannes Bückler, or alias Schinderhannes. He was born in the late 18th century in Germany. He originated from an old family of executors. So it's not a surprise he decided to become a criminal. In fact, he was one of the most known criminals in Germany at the time. He was proven more than 200 crimes, more than 90 accomplices, one of which is Christian Reinhardt, and I would like to mention this person because it plays a role in our story here. The two of them were committing crimes together. Police was trying to catch them for years and finally succeeded in arresting them in 1802 in Mainz, the city of Mainz, which at the time belonged to France. So the investigations took place. They had a court hearing like unobserved at the time with professional judges. More than 400 witnesses were interrogated. And not too much surprise, he and a couple of his accomplices were sentenced to death in 1803. Now, this must have been a particular scenario here, this execution by guillotine. So many people wanted to see it that they needed to move outside the city of Mainz, and it's reported that more than 30,000 people came to see this execution. Even more interesting is that once the execution was done, the heads and the bodies, they were brought to nearby barracks. They were specifically built for that purpose for experiments. Now the experiments were It's a group of professors from the city of Mainz, and they were conducting galvanic and electrical experiments on the heads and the skeletons in order to investigate if they see any emotional reactions. So they were literally putting the heads back to the skeletons, and even more so, they were changing the heads between individuals. I wonder if they got an ethical commission looking at this prior to their study. Anyway, the remains were then brought to the Anatomical Institute in Mainz, and later, when the first chair was filled in Heidelberg, Fidelis Ackermann, he brought the collection of materials for teaching purposes to this institute, including the remains from those individuals. Now, after Fidelis, other subsequent chairs followed, and they modified the collection and they modified the numbering system of the skeletons. We need to remember at this time, the most important thing at an anatomical institute was the collection of skeletons. And here they are. Here are the two skeletons of the two individuals, Reinhardt and Bückler, standing next to each other, and you can judge by the color that these bones have severely been cleaned and macerated in order to be presented in this way. Now, going back to these records, they found some ambiguities in the recordings, which is why they decided to have isotope fingerprints done on the skeletal remains. You can use those in order to infer the geographical provenance of an individual. And the provenance that they found here did not match the records of Bückler. This is why they decided to go for a DNA test. And they wanted to have both cranial and postcranial elements tested for two main questions. First, are the skulls belonging to the skeletons, or were they mixed up while they were changing heads from one body to the other body? And are the skeletons authentic? Now, to answer the second question, we need reference samples. So a genealogist was invited to join the team, Dr. Jan. He was developing the paternal and the maternal genealogy. And what he found is a present-day living person who is maternally related over 5 generations with our person of interest. So they share the same mitochondrial DNA. Now, as samples for extracting DNA, we got 5 samples from Buechler. As you can see, some of the skeletal remains and some of the head. And we got 2 samples from Reinhardt, the tooth and the rib sample. We quantified mitochondrial DNA, 143 base pairs, and you can see, to our surprise, we got quite decent amounts of mitochondrial DNA here, much more than we were expecting, which is why we decided to go for an amplicon-based amplification and sequencing strategy by using the Precision ID mitochondrial panel. And in that case, we went for the whole mitochondrial DNA genome. Now, for all the samples that we were investigating, we got full mitogenomes. So the analysis was extremely successful. We got a haplogroup T2F haplotype for the phalanx of Buechler. And all the remaining samples from the same person got the same mitochondrial DNA, the same haplogroup. So we can, from this result, we know that the skull can belong to the skeletal elements. And the same was true for Reinhardt. The tooth and the rib got the same mitochondrial DNA belonging to haplogroup K1A. Therefore, the skull can belong to the same skeletal remains. Now, what does the maternal reference tell us? We were investigating the mitochondrial genome there, and we found K1A again. Now, we know that this maternal reference is related to Buechler and not related to Reinhardt, which means that we now know that the second skeleton, in fact, is Buechler and not the first skeleton. And this is why we know that for the last couple of decades or even 100 years, the wrong skeleton was assigned to Buechler, and we don't know yet who the second skeleton belongs. And that is still under investigation and research. I was cutting this story very short. It is an interdisciplinary case where we had support from anatomists, anthropologists, radiologists. We had isotope analysis done. We had a genealogist. It was a beautiful, really interdisciplinary team with genetic analysis, for which I'm very thankful in participating. Now, we clarified this case. Let's go to the second case, and you will see soon this is more difficult, particularly in the laboratory, to handle. We stay in Germany, we stay in the 19th century, and we are dealing with the following question. Here is a person where blood and hair samples bring different Sanger sequencing results, mitochondrial DNA. We were asked, can we clarify this with massively parallel sequencing? Who is our person of interest? His name is Kaspar Hauser. He is an interesting person in German history. Unknown is his birth and mysterious his death. The eponym Kaspar Hauser is still used today. We have it in behavioral biology. We see it in medicine and in psychology, in psychoanalysis, and in educational science. And it basically comes back to the fact that a young person, a child, has been socially neglected for a long time. Here is a very brief historical sketch on this person. You can imagine there is a lot of documentation about his life. He appeared out of nowhere in 1828 in the city of Nuremberg in Germany. He was barely able to speak. He was barely able to walk. He reported to have been sitting in a dark room without any social contact for as long as he remembers. He was taken care of by distinguished individuals from Nuremberg, and he was improving very soon. He improved his motor skills, he improved his verbal skills. He became a real celebrity. People would come to Nuremberg just to see him. 5 years after he appeared, he returned home with a stab wound on his chest, of which he died 3 days later. That is the mysterious death of this person. So since then, the origin and the death of this person has been the focus of research. Now, there is a particular element where I would like to spend some time, and that is the Prince theory. At this time, Kaspar Hauser, when Kaspar Hauser was there, the reigning family of the House of Baden was by Karl Friedrich. and he was the Grand Duke since 1806. He had a son, Karl Ludwig, and a grandson, Karl, who was the Grand Duke since 1811, and this is when Karl Friedrich died. Now, Karl and his wife, Stephanie, they had a couple of children, but only one boy. This was a healthy boy when born, but this boy died even before he got a name. Now, since there was no other male descendant in that lineage, the throne went back to his uncle Ludwig since 1818 when Karl died. And in 1830, when Ludwig died, there was no male descendant in this entire lineage, only females, which is why the line of succession changed over to the second wife of the Grand Duke, Karl Friedrich. And this second wife had a son, Leopold, and he was the Grand Duke. So the rumor has it, or the hypothesis is, that actually this family kidnapped the baby, replaced it with a fatally ill baby, and the kidnapped baby is Kaspar Hauser. He was taken away, he was hidden in a place and never saw anybody. So in order to test if that hypothesis could be true, we can DNA test material from Kaspar Hauser and compare it to the mitochondrial DNA of his mother, Stephanie, or one of her descendants in the maternal line. Now, this has been done in 1996. And what you see here is a blood stain on the underwear that the boy was wearing when the stab attack happened. And here you see one of the two living references of the royal family. And as you can easily see here on the screen, they don't share the same mitochondrial DNA. They have very different mitochondrial DNA. Today, with phylogenetic knowledge, we know that the royal family has a lineage called H1BS, whereas the bloodstain on the underwear has a haplogroup that is called W. This was performed by the FSS, Forensic Science Service at the time, and by the Legal Medicine Institute in Munich. Now, of course, that had a big impact on the broad press, but also critics. They questioned the authenticity of the blood on the underwear. They said, look, this has been on museum display, and they may have been tampering with the blood, and this is why we don't see the true mitochondrial DNA anymore. This is why a second round of analysis started using hair samples that were assigned to Kaspar Hauser. They had hair samples antemortem, so when he was alive, and hair samples after he died. At that time, they were pooling hair samples in order to increase the success in getting results, but they got again a different profile. This different profile belongs to haplogroup H. As you can see, it's clearly different from the W profile from the bloodstain, and it is similar to the mitochondrial DNA of the House of Baden. Now, this was never published, but they reported, and that was in line with the recommendations of the International Society for Forensic Genetics, that they cannot exclude the hair samples from coming from the Baden lineage because this is an inconclusive result. So the audience was more or less left with two possibilities. Is the W mitochondrial DNA the one of Kaspar Hauser? Then he would be excluded of being related to the family. Or is the haplogroup H sequence Kaspar Hauser's? Then he could still be related to that family. I'm cutting the story here. There were more analyses done, but they don't contribute to our understanding here, what I'm trying to aim here. The case was reopened in 2019. We were involved in that case. We got 3 envelope of hair samples from Kaspar Hauser. Again, antemortem and postmortem from 2 different families. We selected 4 single hair shafts, and we were analyzing those 4 hair shafts. And as you can see, all 12 hair shafts did not contain any mitochondrial DNA 143 base pair in size. So this is why we moved to a different method, now not based on PCR, but based on capture. This method is called PEG. It was developed from ancient DNA scientists to type the Neanderthal mitochondrial DNA. Now, the PEG method that we used here is not based on PCR, it's a capture method, and I'm going to be talking about this tomorrow. in a presentation that is more technical than the one today. But in a nutshell, we are using human-specific primers to enrich the target, which in our case is human mitochondrial DNA in hair, in order to get better chances of success. And we were surprised how well it worked. Of those 12 hair shafts that are a couple hundred years old, 10 out of the 12 gave reasonable results, some with low coverage, but the majority had good mean read depth. And all the results that we got pointed to the same mitochondrial DNA sequence belonging to this haplogroup W that is identical to the haplotype that was found in the blood sample in 1996. We suggested to involve a third laboratory, and Michi Hofreiter, who is a professor in evolutionary adaptive genomics, and Professor Tori King from Leicester, now moving to Bath, were happy to join our team and try a hair sample. But they didn't know what the case was. So they were involved in the case without knowing what the person of interest was. They used a different method. They used shotgun sequencing, and they arrived at the same haplotype that we found in the other hairs, and that matched the haplotype that was found in the blood sample. Now, we try to address this in a statistical way, and we argue that the analysis performed in Munich on the blood, in Innsbruck on hair, and in Potsdam on hair are three independent experiments. They all lead to the same result. This haplotype is observed 20 times in the restoration part of the MPOP database. We wanted to create a conservative estimate. This is why we added those three haplotypes to the database. We estimated a Klopper-Pearson 99% confidence interval, and the upper limit was 1 in 410 for the probability that a randomly selected person in the population matches this haplogroup W type. Consequently, the probability that the investigation of the sample type in Innsbruck yielded the same sequence as the one in Munich is 1 to 410 if the sequence is not Kaspar Hauser's. And the probability that the analysis of both samples typed in Innsbruck and in Potsdam yielded the same mitochondrial DNA as the one in Munich is a very low number that you can see here if the sequence is not Kaspar Hauser's. So we are providing strong evidence that the haplogroup W haplotype that we found in all the hair samples and in the blood stain is the one from Kaspar Hauser, and that definitely excludes him from belonging to the noble family. Now we want to question, why did the previous hair analysis on Sanger give different results? What we are seeing here is a degradation experiment with short and a bit longer mitochondrial fragments. The short ones correspond to what we typically target with PEG, and the longer ones is what we target with mini PCR, mini amplicon PCR. We know that in hair, mitochondrial DNA is degraded, particularly in old hair. So apparently with the MPS method, we were able to address these short fragments, whereas longer amplification products would not be able to capture this signal. The conclusions are that MPS provides new technical insights into challenged samples. It is amenable to non-PCR-based techniques that can target very short fragment sizes, and that is not possible with Sanger sequencing. But those short fragment sizes may be the only surviving in old and challenged samples, and therefore may be presenting the genuine DNA. We know that modern contamination is typically present in larger fragments. While we try to minimize this kind of contamination, we never can fully exclude it. And the conventional PCR-based sequencing, particularly the longer fragments, they may pick up contamination and miss the genuine signal, which may lead to biased results. So in that respect, MPS is a game changer. Now, I would like to acknowledge the Kaspar Hauser team here. This was a great collaboration from a variety of excellent scientists. Everybody who has participated in this study before has agreed to participate again. We are now able to provide a closing paper to this question that was there for a couple hundred years. It was a collaboration between institutes in England, in Germany, in Austria, and in the United States. This paper is currently under review in iScience, and I'm thanking again this team. So with these two cases, I would like to thank you for your attention. Thanks for listening and be here at HITS. Of course, I'm happy to address any questions that you may have.